They the retail customer.
Is the screen visible to everyone? So there are few participants who have unmuted themselves. So kindly mute yourselves. Okay. So let's start with
Students, I
today's
request
paper.
all of you kindly put your mic in the mute mode when ma'am is taking the class. Don't disturb. If you don't want to do the class, then don't do. If you have a doubt, if you have a query, you can ask. But don't just put my mic on mute and you ask. Fine? Thank you. Go ahead, ma'am.
Okay, so let's see what we have today. So today we will be discussing about the third block that deals with your sex determination, sex-linked inheritance, some of the genetic and chromosomal abnormalities found in human beings, and genetic analysis of quantitative or polygenic traits. So let's start with the first slide regarding the sex determination. So if we talk about the topic, this is actually sex determination means where a process or method is used for predicting or detecting the sex of an individual. So primarily we have two categories of sex, male and female. So the process or the method that is used to find out okay. First, let's continue today's class, and whatever questions or queries you have, you can discuss at the end of the class. Okay. So, sex determination, as I was telling, this is the way, or this is a method or procedure by which you can predict or you can determine the sex or the gender of the individual. Okay. So, primarily to find out whether an organism will develop into a male or a female, that process will be known as sex determination. So broadly, there are two different ways or mechanism of determining the sex. The first one is the environmental mode of sex determination. The second one is the genotypic mode of sex determination. So as the name indicates, the environmental mode, in this environmental mode of sex determination, the environmental factor will determine whether a zygote or the offspring will develop into a male or female. So environmental factors like your temperature, the location, okay, etc. So these will be determining whether an individual will develop as a male or will develop as a female. Now, in the genotypic mode, as the name suggests, the genetic makeup will determine whether the individual will develop into a male or to a female. So let's see some examples like here. You can see that the three different graphs represent how the temperature is responsible for the determination of the sex in the individuals. So in the first graph, if you observe, this y-axis is indicating the percentage of female and the x-axis is indicating the increase in temperature. So in the first case, if you observe, when the temperature is low, the percentage of female is 100. That means if the eggs will be hatching in a low temperature environment, all the offspring will grow into a females. Now, as gradually the temperature is increasing, you can observe there is a shifting in the percentage of females. That means in the high temperature, the fertilized eggs will be developing into males. And this is indicating a transitional state where there will be 1:1 sex ratio. That means at this particular mid temperature, 50% of the eggs will develop as female, 50% will be developing as males. Now here in the second graph, it is quite opposite. Okay, so if the temperature is high, then the fertilized eggs will develop into female, and the low temperature, the fertilized egg will be developing into males. Now here, if you observe, there is a means little bit complex thing is over here. So at low temperature, at extremely low temperature, and at extremely high temperature, the eggs will be developing into females, and in an intermediate temperature, the eggs will develop into male. So this is how the temperature, the environmental factor is responsible for determining the sex of the individual. Now, if we look into the genotypic mode, that is most common in many of the higher group of organisms, even including human beings also. So we have this XY method of sex determination. So in our case of humans, the females are homogametic, means they will be having 2 copies of the X chromosome. And the males are heterogametic, that means they will be having 1 X chromosome and they will have 1 Y chromosome. So in case of human beings, the Y is actually the principal chromosome for determining the sex of the individual, or it actually accounts for the maleness. The absence of Y will be responsible for the femaleness. Okay. Now, it's not universal that in all the animals, the female will be always homogametic and the male will be heterogametic. In contrasting to this, particularly in some group of birds, if you observe, it is reverse. That means in birds, there is ZW method of sex determination where the female is heterogametic. That means the female is ZW and the male is homogametic. That means the male will be having Z and Z. Okay. Then coming to the C. elegans. So this is a means worm. Okay. So here this is also has a quite interesting pattern of sex determination. So now in this particular organism, there is no male and female, rather there is a hermaphrodite and there is a male. So hermaphrodite means an individual having both the male and female sex organs. So in case of C. elegans, either the organism will develop into a hermaphrodite or it will develop as a male. So usually the worm having 2 X chromosomes will develop as a hermaphrodite, and the individual or organism who does not have 2 X chromosome or there is a missing of X chromosome, it will develop as a male. Okay. Now again, how this is particularly determined? Now this is determined based on the fertilization status. Okay. So there could be 2 possibilities. Either since the hermaphrodite has both male and female sex organs, it might go for a self-fertilization. So self-fertilization means the ovum and the spermatozoa will be produced by the same individual. So if there is a self-fertilization, then all the fertilized egg will develop into hermaphrodite. Okay. But if there is a cross fertilization, cross means different. Okay. So here the egg will be released by the hermaphrodite and the spermatozoa will be given by the male individual. So if there is a possibility of cross fertilization, the cross fertilization will generate the hermaphrodite and in the male in a ratio of 1:1. That means 50% of the eggs will develop as hermaphrodite. The rest of the 50% will develop as the males. Then this is the normal karyotype of a human being. So based on the sex chromosomes or allosomes, we are differentiating them as male and female. So if there are two homogametic chromosomes or two homologous X chromosomes are there, then it is considered as a female. A heterologous pair of X and Y is considered as a male. Now, this is the Y chromosome, which is responsible for the maleness in case of human beings, particularly not only human, in most of the mammals actually. So, if we look into the segments or the portions of the X chromosome, as you can observe, there is a pseudo-autosomal region. So, pseudo-autosomal region means this is actually a sex chromosome or allosome, but some part of the chromosome has resemblance with that of the autosomes. Then this is actually the most important segment or gene that is responsible for the sex determination or the means development of the maleness, that is the SRY, which is the sex determining region Y. Now this is the centromere, this is the euchromatin region. So euchromatin means it is actually having genes that are expressible. Then this is the heterochromatin region, which in which there are genes which are permanently switched off. Okay, and this entire segment is consisting of the male specific region Y. Okay, so sometimes you can observe that this pair can have homology with that of the X chromosome of the female as well. But this specific region is quite unique to the Y chromosome. This male specific region Y, which includes the euchromatin, the heterochromatin region, the centromere. These are the characteristics of the Y chromosome. In addition to this, the SRY, the sex determination region on Y chromosome, that is highly responsible for determining the male and the female. So how exactly it happens? What is the mechanism behind this? So if we look into the developing zygote, so before the individual develops as a male or female, it is undifferentiated. The gonad is undifferentiated. Now there is a means transcription factor which is called as the testis determining factor, the TDF. So if the TDF is actually means coded by the SRY region, okay. So if the Y chromosome is present, then what will happen? There will be testis determining factor will be released. So if the testis determining factor is released, it will help in development of the testis. Now this will again promote the presence of MIF. So MIF is your Mullerian inhibitory factor. So the presence of MIF will help in regression of the Mullerian duct. Okay. So Mullerian duct, they are actually the female reproductive ducts like your that usually gives rise to the future oviduct, that is the fallopian tube, etc. So if the testis is developing, the testis will be promoting the secretion of the MIF, and the MIF will inhibit the development of the female specific gonadal ducts, and the presence of testosterone. The testis will also secrete the testosterone. The testosterone will develop the growth of other accessory sexual organs like your seminal vesicle, vas deferens, etc. And this is also another compound, that is dihydroxytestosterone. This will also promote the labial scrotal fold, the growth of phallus and the prostate. So all of these are actually accessory male reproductive organs. But what will happen if there is no Y chromosome? So if there is no Y chromosome means there is no testis determining factor. So in absence of testis determining factor, the gonad will differentiate into ovary, which is the primary female sex organ. Now the ovary will be secreting the inhibitory MIF, that is the Mullerian inhibitory factor, which will prevent the which will promote the differentiation and growth of the fallopian tube, uterus, the upper 1/3 part of the vagina, etc. And the testosterone that is produced by the ovary will be actually helping in regression of the Wolffian dogs. Okay, actually it should be not testosterone, this should be estrogen. Okay, so this will actually inhibit the regression of the Wolffian duct. So Wolffian duct means the duct that is actually giving rise to the future seminal vesicle, vas efferentia, vas deferens, etc. That is the male accessory reproductive ducts. So the regression of the Wolffian duct will be leading to development of the entire female reproductive structure. So that is how the undifferentiated gonad will develop into either testis or ovary because of the presence or absence of the testis determining factor, which is actually encoded by the sex determining region on Y chromosome, that is the SRY gene of the Y chromosome in humans. Now, coming to the sex determination in Drosophila. So, this is popularly known as the genic balance theory. So, here, although the Y chromosome is usually present in the male, but Y chromosome does not solely determine the sex of the individual. So, there is a balance, the balance between the X chromosome and the autosome will determine what the individual will develop into. So, if the ratio of the sex chromosome and autosome is 1, it will develop as a female. If the ratio of the autosome, sex chromosome and autosome is 0.5 or 1 by 2, then it will develop as a male. And if it is more than 1, it will develop as a meta female, or sometimes also called as a super female. And if it is less than 0.5, it will develop into a meta male, that is a super male. Now, if it falls somewhere between 0.5 to 1, then these kind of flies will be referred to as intersex. Okay. So the meta female and the meta male, or the super female and the super male, they are actually highly unstable, and usually the flies with this kind of genetic makeup, they die. They are not able to survive. And the intersex, they will survive, but they will be sterile. Okay. So sometimes what you will observe in a particular fly, there might be presence of a Y chromosome, but still the fly will be considered as a female. So why? Now because of this ratio of the X chromosome and the autosome. Now you may question if there is presence of Y chromosome and it is developing into female. So although it will develop into a female, but it will not be able to reproduce as a male. So it will remain as a female. Now in some cases what you might observe, depending upon the ratio of the X by A, there will be no Y chromosome, but still the individual will be regarded as a male. For example, you can look into this combination. Okay, so here look into the genotype. There is no Y chromosome, but since the ratio is 0.5, it will develop into a male. So although it will develop into a male, but because of the absence of Y chromosome, it will be a sterile male. It cannot reproduce. Okay. Unlike here, if you observe also, there is presence of Y chromosome, but still it is developing as a female based on the genic balance theory. So although it will be a female, but it will be again sterile in nature because of the presence of Y chromosome in the female. So how exactly the male and the female development takes place in the Drosophila development? If you remember, yesterday we discussed something called as the alternative splicing, right? I think I showed you the example also. If there is a means the mature polypeptide will be containing exon 3 and 4, it will be developing into a male. And if the exon 3 and 5 are joined, then it will develop into a female. Okay. So, what happens during the gonadial development? If the X by A ratio is 1, it actually leads to expression of a protein called as SXA. Right? So that is actually sex induced lethal gene. This is called as SXL, okay, or sex specific lethal gene. So if this SXL is means it is produced, or if it is activated, it actually leads to the splicing of this mRNA. Okay, the mRNA that is called as the TRA, and this mRNA will again go and induce another mRNA, that is TRA2. So the TRA and TRA2 together will promote the expression of a female specific protein, that is the DSX. Okay. So this is called double sex protein. Now, if the ratio is 0.5, so in this case, what will happen? This SXL gene will not be produced, or this protein will not be produced. So if this protein is not produced, the TRA will not be, means it will not be transcribed, and in absence of TRA, only the TRA2 protein will be produced, and the TRA2 protein will improvise, or it will actually promote the synthesis of male specific protein, male specific DSX protein. So that is how the ratio will be determining the expression profiling of the genes, and accordingly the individual will develop into a male or to a female. Now there is a thing called as dosage compensation. So dosage compensation means, since there is an imbalance of the X chromosome or the sex chromosomes in male and females, so multiple mechanisms have been adopted to balance the expression profiling of the X chromosome in males and females. So one way is the random inactivation of the one of the X chromosome. Like in case of human beings, you observe the females are having two X chromosome, the male is having a single X chromosome. So what happens in case of human beings? One of the X chromosome is deactivated or inactivated, so that the X number is balanced in both male and female. That is called random inactivation of the one of the female X chromosome. Okay. Then in other cases, such as your Drosophila, in Drosophila also usually the X chromosome in males will be half the number of the X chromosome in the female. So in that case, what is happening? There is a two-fold transcription of the single X chromosome in the male. So that is called as the hyperactivation. So in that case, whatever a single X chromosome is present in the male, that will be transcribed two times, or the expression will be two times, which is same as two X chromosomes that is present in the female. So this is called hyperactivation. Now, in case of C. elegans, what happens? In C. elegans, the female is having two X chromosome and the male is having one X chromosome. But here in the female, the expression of the X chromosome is actually reduced, okay, to half. So that is why half will be equal to 1, which will be same as the one single X chromosome in the male. So this is called decreased transcription or hypoactivation of the X chromosome in the female. So three different ways of dosage compensation. In case of human beings, it is random inactivation. In case of Drosophila, hyperactivation of the single X chromosome in the male, and in case of C. elegans, it is hypoactivation of the two X chromosomes in the female. So this is the how exactly this is the molecular mechanism how the dosage compensation is done in case of Drosophila. So there is another protein called as the MSL2 that is responsible for the hypo or hyperactivation of the X chromosome in case of your male Drosophila. So in case of males, since the SXL is not produced, okay, so there will be sexual differentiation of the male sex organs. But due to absence of the SXL, the MSL2 will be activated, and due to presence of the MSL2, this will promote the dosage compensation. That means it will help in increasing the expression of the single X chromosome which is present in the male. Now, what will happen in case of females? Since in case of females, the SXL is produced, the SXL is actually inactivating this MSL2. So, since the MSL2 is not present, there will be no dosage compensation. Now, coming to the example of dosage compensation in human beings. So in human beings, it is by random inactivation of the one of the X chromosomes in the female actually. So if you see in the these are the somatic cells of the male and the female. So usually in the female somatic cells of the female, since one of the X chromosome is inactivated, inside the nucleus you will find one X chromosome attached, inactivated X chromosome attached to the nuclear membrane like this. Okay. So this the inactivated X chromosome is known as a bar body. So in a genetically normal female, there will be only one bar body found in the somatic cells. But if there is certain kind of genetic abnormality like in Klinefelter syndrome, so in case of male, there should not be any bar body. There is only one bar body in female, no bar body in male. But if there is certain kind of genetic abnormalities like the Klinefelter syndrome, so in that case, it will develop as the male can also have a single bar body. Okay. And if there is any female is born with genetic abnormality like more than 2X chromosome, then you can have more than two bar bodies in case of human beings, human females. Now, this X inactivation in females is actually explained by a hypothesis called as the Lion hypothesis. So to further explain the X chromosome inactivation, the female mice heterozygous for the X-linked coat-colored gene were studied. Based on their study, Mary Lyon and Lyon Russell, they made independent hypothesis in 1961. So what were the major points or the postulates of the hypothesis? Now, the females have two X chromosomes. One is the paternal and the other one is the maternal. During early development, there is inactivation of one of the X chromosomes in the female somatic cells. So, every human female has inherited one X chromosome from their father and one X chromosome from their mother. So, this is called as the maternal chromosome. This is called as the paternal chromosome. And during the development, one of the X chromosome is actually inactivated. So out of the two X chromosome in females, any X chromosome randomly becomes inactivated except in marsupial and in some extra embryonic tissue of some placental mammals. So usually the X inactivation is a random process. That means whether the paternal X chromosome will be silenced or the maternal X chromosome will be silenced, it is completely random. That means during the development, the cell randomly can either silence the paternal chromosome or can silence the maternal chromosome. Now, once the X chromosome is inactivated in progenitor cells, the same X chromosome will be inactivated in all the descendant cells. So, this is actually important to remember. If, for example, if this is the cell where the paternal chromosome is inactivated, so which X is active here? The maternal. So whatever daughter cells will be produced from this all will have the silenced paternal chromosome and all will have the active maternal chromosome. Okay. Now, similarly, if in another cell the paternal chromosome is active and the maternal chromosome is silenced, then in all the cells, the maternal chromosome will be silenced and the paternal chromosome will remain active, like this. Okay. So here the orange is representing the maternal chromosome, the black is representing the paternal chromosome. So like if you see here, in whatever cells the maternal chromosome is active, all the daughter cells produced from that cell will have the maternal active chromosome, and all the daughter cells where the X chromosome is active will have the paternal X chromosome is active will have the paternal active X chromosome. So now how exactly one of the X chromosome is inactivated? So we have yesterday also we discussed that RNA concept, the interference RNA. So similarly here there is a transcript called as the cyst. Okay, this is actually X inactive specific transcript. So this if there is the development is going on, so randomly one cell is chosen, and in that cell what will happen, the gist will be made on, and this TSIX, that is actually which is antisense to the gist, so that is actually off, because in other cases if this is active it will bind to this and this cannot be functional. So if the gist is operating means this TIS has to be off. Okay, so now the 6th RNA will be accumulating and they will go and they will bind to the X chromosome. So if they will go and bind to the X chromosome, that means the X chromosome can no longer, means it will always remain in a permanently off position. That means it is inactivated. And whatever X chromosome has to remain active, in that case, this antisense gist will be active. So the antisense gist will be bound to the gist so that it cannot act on the silencing of the X chromosome. So in that case, the chromosome, the X will remain active. Now this is in brief about how the sex is determined and how the sex chromosome expression is balanced in both the sexes by doses compensation. Now next we will go into the study of human genetics and some kind of chromosomal abnormalities. So there are various methods or techniques by which you can study the genotyping of the human beings. One such method is called as the preparation of the karyotype. Okay. So karyotype, as you can observe here, is actually the arrangement of the entire chromosome for visualization. Okay, so this actually arrangement of the chromosomes in the for visualization and analysis for basically structural and any kind of numerical aberration is called as karyotyping. So this is the procedure of karyotyping. So 5 ml of venous blood can be extracted and the compounds such as your phytohemagglutinin and culture medium is added, or the blood is actually used in the culture medium like at around room temperature for 3 days. Then colchicine and hypotonic saline are added. So colchicine is added to actually inhibit the cell growth, okay, or to inhibit the cell division during the metaphase. um, then the cells are fixed. So once the cells are fixed, they can be taken out and they can be spreaded into a slide by simple dropping, and they are digested with the proteinase enzyme called as the trypsin, and they can be stained with a stain called as the Giemsa stain. Now, then they are ready for visualization. So you can directly observe them under the microscope, and they are usually arranged as per their size, decreasing order or descending order, we can say, from largest to the smallest. And usually the sex chromosomes are placed towards the end. So that is how the karyotyping is done. And by looking into the karyotype, you can easily find out if there are any kind of structural abnormalities, like if there is any deletion, there is any inversion or duplication of the chromosome. Then there are other techniques also. So few other techniques will include like the banding techniques. Okay, so we can have or we can actually stain the chromosomes, and as per the sequence or the nucleotide sequence that is present within the chromosome, they will be taking a different differential amount of the strain. Differential amount of the stain. So usually the AT regions, they have different type of staining, and the GC region will have different type of staining. Then how much stain they are absorbing, based on that we can predict the structure and the distribution pattern within the chromosome. So just let me show you one diagram first, then it will be easier for you to graph. See, like this. Once you stain the chromosomes, you can have banding patterns like this. Okay. And every chromosome will have a unique banding pattern, because every chromosome has different set of gene or different sequence of DNA or different arrangement of AT and GC regions. So that is how they will be taking different type of staining in either AT or GC rich region, and accordingly you will have a fixed banding pattern for the entire set of chromosome that is present in human beings. So this is the chromosome during the prophase, so which is actually a little bit less compact phase. And as the chromosome will be more compact in the metaphase, the banding pattern will slightly change. But for a particular stage of the cell cycle, the banding pattern will remain fixed. So this is the chromosome banding pattern in chromosome 1, and this is the banding pattern of the X chromosome. Now, in every kind of karyotype, there will be exactly two chromosomes having this exactly identical banding pattern. So that's how we will be identifying the homologous chromosomes from them. So there are different types of banding techniques are there, like Q or quinacrine banding is there. Then there is G or GIMSA banding is there. Then there is C or constitutive heterochromatin banding is there. Then the R or the reverse banding is there. So let me explain just the GIMSA, which is most commonly used in cytogenetics. So the G banding is carried out by treating the chromosomes with the protease enzyme trypsin to digest the chromosomal proteins, that is the histones, etc., that is associated with the chromosome, followed by staining with the GIMSA dye, which is a combination of two compounds, that is thiazine and eosin. So the preparation is examined under a light microscope. The dark G bands represent the AT region, the late replicating heterochromatin, which is generally inactive, and the light bands are indicative of generally active, oddly replicating, that is the GC or U chromatin region. So advantage of gene sustaining are that it produces permanent preparations which can be visualized anytime without the loss of clarity, and the chromosomal structure changes can be identified accurately under a light microscope. Okay. So let me tell you what exactly it depicts. So if you look into this particular chromosome, you can observe there are two types of bands are there. One is thick or dark band, the other one is the lighter band. Okay, so the dark band is actually representing the AT rich region, means in this particular region the DNA sequence contains most of the bases that are adenine and thymine. And the light, so and this is actually indication of heterochromatin region. So heterochromatin means the genes from this region are usually not transcribed. Okay, or we can say there is compact arrangement of the nucleosomes in this particular region, that is why there is no expression of the gene from this region. But the lighter regions, what you see here, okay, so that is indicating a GC rich region. So that means the guanine and cytosine region. Okay. So in that particular region, there is means it is more flexible. Mostly the genes are active. That means they are transcribed into mRNA from this region. So this is actually called as a euchromatin region. And most of the times, if you observe, the centromere is actually mostly heterochromatin in region, because most of the genes that is, or most of the sequence of DNA that is present in the centromere, they are permanently inactive. Okay, so usually the centromere will be representing a constitutive heterochromatin region. Now coming to the nomenclature of the chromosome bands. Okay. So how we will be identifying that there is some problem in the chromosome or not. So before that, we should know how the structure of the chromosome is described. Okay. So we can have four different type of chromosome based on their structure, or we can say based on the position of the centromere more accurately. So in some cases, like in the first case, you can see the centromere is present over here, and the two sides of the chromosome are very unequal. Like these are called as the arms of the chromosome. So in case of acrocentric chromosome, one of the arm is very long and the other one is very short. Then there is a slight increase in the length of the short arm. So this is called a submetacentric chromosome. Okay. And this metacentric chromosome, usually both the arms are of equal length or almost equal length. And in telocentric, there is like the short arm is completely absent and the centromere is at the end of the long arms. So the short arm is actually called as the P arm, P actually it is derived from the word petit in Latin, which means small. And in contrasting to this, the long arm is called as a Q arm. Okay. So how we can name or how we can find the location of a gene in the chromosome, if you observe. So this is the P because this is the short arm, and this is the long arm, that is the Q, and within the P there are two broad banding regions are there, 1 and 2, and within the 2 region again there is one T rich region, one GC rich region, so they are called as the sub band, or sorry, bands, and again within the band there could be sub bands. Okay, so like if you observe here, this is given 15.1, so this means it is actually, it will be written by P 15.1. So what does it mean? Now this is telling about a location on the chromosome which is present on, actually the chromosome number is actually mentioned before this, like 1P151. So this is actually present in the first chromosome, in the short arm of the first chromosome, in the first region of the fifth band of the first subband. Okay, so that is how the location of a gene is revealed from the banding pattern of the gene. Then comes another technique called as the flow cytometry. So this technique actually involves the sorting of the chromosomes based on the apply of the fluorescence dye. Okay. So the instrument is called flow cytometer, and the technique is called as the flow cytometry. And the process by which the sorting takes place is called as FACS, fluorescence activated cell sorting, FACS. Okay. So as you can see here, one of the chromosome is labeled with a fluorescent dye. I hope everyone understand what is meant by fluorescence. So fluorescence means a particular color. Okay. So one of the chromosome is stained with a fluorescence dye. Now, when it is put in that machine or the flow chamber, so there is a light source which will actually help in emission of the fluorescence from this particular chromosome. Then there are fluorescence detectors are used, and as you can observe here, there are deflection plates are there. Okay, so as the deflection plates, they are charged with particular ions. Okay, so this is a positively charged plate, this is a negatively charged plate. So now so here you can actually change the charging signal, means depending upon which chromosome you want to short, you can change the charge accordingly. Okay, so here actually we are targeting the chromosomes which are having a positive charge, so it will be deflected towards the negative deflector plate, and they will be collected in the fluorescent tube or in the collection tube. And whatever chromosome they do not have our desired charge or our desired fluorescence probe, they will simply go to the waste. And after that, when you analyze, you will get that all the chromosomes you had labeled with the fluorescence dye, they have been collected and they are now sorted. So again, what is the utility of this procedure? Now, this can be used to identify certain kind of genetic abnormality, like if there is any kind of structural aberration or numerical aberration in the chromosome. Then there is another technique called as the FISH or fluorescence in situ hybridization. So here what happens, instead of labeling the entire chromosome, a part of a probe is used. Actually, a fluorescence probe is used, means you have a segment of DNA that is labeled with a fluorescence dye. Okay, and you are releasing it into the cell, or you are releasing it to bind with the other chromosomes that is available, or the sample chromosomes. So what this probe will do, this probe will go and it will try to find a complementary sequence that is present inside the chromosome. So wherever it will find the complementary sequence, the fluorescence tagged probe will go and bind, and this process is called as the hybridization. And before that, usually it is means they are unwinded, or it is actually what we can say, the chromosome from has been melted from double stranded to single stranded. Okay, so or we can say it is denatured. The DNA has been denatured and the single strands have been exposed, so that the fluorescence tagged complementary probe can go and bind to the complementary site on the target chromosome. So wherever the fluorescence tagged probe will bind, it will give an emission or it will give a color that can be easily visualized and analyzed under a fluorescence microscope. Then there is another technique called the spectral karyotyping, like this. Okay, so here this is actually more interesting, because what you are doing here is you are using 24 different type of fluorescence label tags for 24 types of human chromosomes. So we have 23 pairs of autosomes, okay, and one pair of sex chromosomes, that is either XX or XY. So how many different types of chromosomes are there? So there will be, 22 pairs of autosome, 22 pairs of autosome and one pair of sex chromosome. So the sex chromosome is again two types, that is X and Y. So altogether there are 24 different types of chromosomes are there. So by using flow cytometry, if you can identify, or if you have sorted, or if you have separated all the 24 chromosomes in different tubes, then you can label them or you can color them with 24 different types of fluorescent markers. Okay, and then you can analyze or visualize them. Each color will be representing a set of chromosome or a particular chromosome, and every chromosome will be present two times, so there will be two chromosomes from each color. Page number, I don't understand what do you want to ask. Page number, or you are asking the slide number? I think now I am on slide number 33. Okay, in material, actually, I'm so sorry, I don't know in which page number material it is present. Okay, it is 43. As per your friend's suggestion, it is 43. Okay, can go to the page number 43. Now, there is another technique called as CMA, that is chromosomal microarray analysis. So this is also another technique which is used to predict if there is any kind of structural aberration in the chromosome. So as you can observe here, this is how the microarray analysis chip will look like. So you can observe there are three different types of colors are there. One is yellow, the other one is green, and the third one is red. Okay. So here what is done, two different DNA have been mixed. Okay, one is the patient DNA, that is our sample, and the other one is the reference DNA. Reference means a standard DNA or a control DNA, which is having normal copy. Okay, that means there is no alteration or no aberration in the reference DNA. So what will happen if you mix them and if you allow them to hybridize? Now you can observe different color patterns here. So what could be the possible things? Now the patient sample or the sample that you are using, it could have a normal DNA. That means there is no change in the chromosome. So in that case, control will also have two copies. This patient with DNA or the sample DNA will also have two copies, and the ratio of the green is to red will be 1:1, and that is how you will get a balanced color, that is yellow color. But if there is some kind of duplication, okay, or multiplication or abnormal number of chromosomes in case of your patient sample or sample that you are analyzing or detecting, then what will happen? The green color will be more, okay, because after neutralizing the red color, there will be excess of green emission. So the green is representing some kind of abnormality. Okay. Now, the in case of red, what will happen now if there is some deletion? So in deletion also, that means there is lack of normal DNA copy. So in that case, the green emission will be less, and since the control is having two copies, they will be reflected more. So you will have the red color emission like this. So both red and green are indication of gain or loss of chromosome. Green is an indication of gain of chromosome, that is duplication, and red is an indication of the loss of chromosome, that is deletion. Only the yellow samples will be the normal ones. Okay. So this is called as microarray analysis, and this is very useful because means we can say 100 of samples can be simultaneously analyzed by this technique. Then coming to the some numerical aberrations in the chromosome, which is called as the aneuploidy. So there are two types of numerical aberration in chromosome number. So one is aneuploidy, another is polyploidy. So aneuploidy is when there is increase in decrease of single number of chromosome or individual chromosome numbers. And polyploidy is when the entire set of chromosome number will be changed. Like for example, we are having 2n or 46 number of chromosome. Okay. Or so this is the diploid case in our condition. Now what is the haploid condition in our case? That is 23. So if a cell will have all the 23 in triplicates or all the chromosomes in triplicate, okay, so that means the total number of chromosome will be 69 and every chromosome will be present in 3 times. So that is a condition of polyploidy where the entire set of chromosome is duplicated. Now in aneuploidy, what is happening? Now there is a change in number of individual chromosomes. Okay, like if we consider like 47 chromosomes are there. So all the chromosomes are in duplicates or in diploid condition except the X chromosome where it is XXY. So there is a increase in number of X chromosome. Single X chromosome has increased. So this condition is called as your aneuploidy. So how exactly the aneuploids are developed during the meiotic process or meiosis? Now that actually occurs because of a condition called as a non-disjunction of homologous chromosomes. So there are two possibilities. I hope all of you remember the meiosis, how meiosis happens. So there could be two possible ways of non-disjunction. The first case, non-disjunction during the meiosis 1, and the second case, non-disjunction in the meiosis 2. So if you remember in the first meiosis or in the meiosis 1, what happens? Simply the homologous chromosomes are separated. Means if we have chromosome number 1 like this, or let's take the example of the X chromosome. Okay. In case of a female, let's say. Okay, so the female is having 2 X chromosomes. So in the first meiosis, both the X chromosome will move to 2 different cells. Okay. And then they will split and the sister chromatids will be produced, and each of the gametes will be receiving half of the sister chromatids. Like this. So this is the normal condition. But if these two homologous chromosome or these two X chromosome which were supposed to move to different poles will not separate, then what will happen? Now both the X chromosome will move to the one cell and the other cell will not have any X chromosome. Now in meiosis 2, there is normal disjunction. That means the chromatids will be separated. Okay. But now, what is the problem that you can observe? Instead of having one X chromosome, both the cells will have 2 X chromosomes. Okay. So that is aneuploidy condition. And if they are fused with a normal gamete, this will lead to a trisomy condition because this X chromosome is now present in triplicates. Okay. Now, what is happening to the other cell here, since there is no X chromosome? Or let's consider it as the chromosome number 21. Okay, let's consider it as chromosome number 21. This will be easier to understand. So here, this cell is having already 221 chromosomes means it is already diploid for 21. Again, when it will fuse with a gamete that is having another 21 chromosome, so there will be 321 chromosomes in this cell. So that is a trisomic or abnormal condition. Similarly, here the 21 chromosome is completely absent. So this zygote will receive only one gamete from the normal parent, and it will be a monozomic condition. Okay. Now, similarly, sometimes the non-disjunction can occur in meiosis 2. So here what is happening? Normally the homologous X chromosome have separated, and in during the second meiotic division, the sister chromatids, they did not separate. So what will happen again? One of the cells will receive 2 copies of the 21st chromosome, and one of the cells will not have any 21st chromosome. So this will be trisomic, and this will be monozomic after the fertilization. So actually the non-disjunction at meiosis 1 is more dangerous because it will produce all 4 aneuploid gametes. Whereas the non-disjunction in meiosis 2 is less dangerous because at least 50% of the gametes are normal. Then these are the chromosomal structural aberrations that we already discussed yesterday. Like there will be deletions, there will be duplications, there will be inversions, both paracentric and pericentric. Then sometimes the chromosome can means join like this. The telomeres can fuse together forming a ring chromosome. Then there is translocation, etc. So this is an example of a fish that is showing the chromosomal aberrations. Like you can observe here, in the interstitial deletion here in the chromosome number 15, you can observe this green part is actually showing where the chromosome has been deleted. Okay, from this section, this part, or from this portion, few chromosomes has been deleted. And here you can observe there are many different type of this is a cell, or this is a photograph of a cell taken from a bladder cancer. Okay, so here many abnormal chromosomes can be seen. Like for example, here if you see, there is no centromere is present, and sometimes somewhere you can see here also deletions are there. Okay, and in some cases you can observe this is, so this is some duplications can be seen here, and here you can say translocations are seen here. Okay, so many kind of chromosomal abnormalities can be seen. Then coming to so these are the structural aberration that can take place in a chromosome. So there are other external agents also which can lead to chromosomal abnormality, like the ionizing radiation, some chemicals, then some transposable elements, some viruses, the errors during the DNA replication and repair mechanism, then the VDJ recombination and the class switch recombination in lymphocyte, that is primarily in the B and the T cells of the B and T immune cells. Okay, then some fragile sites in the chromosome. So in the chromosome, few sites or locations are there which are very prone to mutation. So multiple times the mutations will occur there, and those sites can break. So if the breakage will occur, that will lead to deletion. So these are the effect of the ionizing radiations that you can observe here. It has sometimes it can form a dicentric chromosome like this, means 2 centromeres on the same chromosome, that is highly unstable. Sometimes it can lead to formation of a ring chromosome, or simply it can induce double strand breaks, and part of both the chromosomes can be deleted and fragments can be seen like this. Now, what kind of chromosomal changes can be induced by the transposons? Okay, so the transposons, these are actually the homologous segments. So the transposons can induce all 4 types of chromosomal aberration. Like they can induce deletion also, they can induce inversion, they can induce insertion, and also they can induce the translocation. So how exactly it is done? If we look carefully, so in the first case, you can observe that there is a deletion of the segment of the chromosome. Okay, so this is the normal chromosome that is having WYZ, and these are the transposons both the terminal sites. So some, since these transpositions are homologous in nature, they can come side by side like this, and during this they can undergo a crossing over process. So a crossing over means part of this will be shifted to here, and part of this will be shifted to here. Okay, there will be physical exchange. So after the crossing over is complete, what you can observe, this is now the chromosome where the WYZ has been deleted. Okay, so this can induce deletion. Similarly, here also the chromosome, the homologous regions can form a loop-like structure like this, NRT. Okay, and there could be crossing over at any point, let's say between these two. Okay, so now the parts has been exchanged, and when it will straighten up, you can observe that the W and Z, they have been interchanged. So this has induced an inversion. Similarly here, this is a ring chromosome which is having this transposition, and when this transposition will come in close contact with another transposition or a homologous transposition, then again there could be crossing over, and because of the crossover, this entire segment can be inserted in between this. Okay, so that is again another chromosomal abnormality. Now here, between two different chromosomes, if the transpositions are located, simply they can undergo crossing over, and they will be producing translocation like this. Okay, so A, B, C, D were part of the chromosome 1, and V, W, Y, Z were the part of chromosome 2. Now the CD has been translocated from 1 to 2, and the YZ has been translocated from 2 to 1. So these are the ways by which the transposable elements can induce certain kind of chromosomal aberrations. So these are the fragile sites on chromosome, which I have already told you that certain regions on the chromosome, they are very prone to mutations. So multiple times, if there will be mutations, so they will be susceptible to breakage. Means usually in the X chromosome it occurs. Okay, so in the X chromosome, the as you can see here, the fragile X chromosome syndrome is there, the fragile X syndrome. So some regions on the X chromosome, they are highly prone to radiations and mutations. So multiple times the radiations and mutations can cause a disruption or the breakage in the chromosome, leading to deletion. Then certain type of abnormalities in the that arises due to trisomy, monosomy, etc. So there are three major trisomies seen in case of human beings: trisomy 21, 13, and 18. Or you can remember in this order: PED, paired, 13, 18, and 21. Okay. So P stands for Patau syndrome, E stands for Edwards syndrome, D stands for Down syndrome. Now, in addition to that, there are two trisomy of the sex chromosome. There is one trisomy of the sex chromosome, that is Klinefelter syndrome, addition of one extra X chromosome. And there is monosomy of the X chromosome, that is Turner syndrome, that is absence of one X chromosome, X0. Okay. So the Klinefelter syndrome occurs in male, and the Turner syndrome occurs in case of female. And these three are the autosomal aneuploid condition which can occur both in male and female. So these are the consequences or the symptoms of the syndrome. As you can see, a lot of abnormal conditions will develop if there is a trisomy X. So, trisomy 21. You can see the karyotype of the individual. So instead of 21st chromosome, there are 3 copies. So it might have occurred due to the non-disjunction of the 21st chromosome in either of the parent, either in the male parent or in the female parent. And if a child is born with this abnormality, then all this type of problem will be present in the fetus. So that is why the prenatal screening is done, to ensure that the child does not have any kind of chromosomal abnormality. Similarly, this is the condition with the Patau syndrome. The chromosome number 13 is present in 3 copies instead of 2, and these are the abnormal condition that will develop due to Patau syndrome. Now, this is again another Edwards syndrome due to trisomy of chromosome number 18. So you just have to remember the number of the, means which chromosome is associated with which syndrome, and what are the different type of abnormalities, the structural and the physiological as well as anatomical abnormality associated with the syndrome. And this is the Turner syndrome. So remember, always Turner syndrome will occur in a female. The female that does not have X chromosome will be suffering from Turner syndrome. So these are the abnormality. Mostly such type of females will be sterile, means they will have rudimentary ovary, because of which they cannot produce the ovum. Then the Klinefelter syndrome. In the Klinefelter syndrome, this is the presence of extra X chromosome. Okay. So this will always occur in the males. So due to presence of one X chromosome, it might have certain kind of female characteristic development like the gynecomastia, that is presence or development of the breast in case of males, rudimentary testes, and development of certain female reproductive organ might take place. And such type of males will be also sterile in nature. So these were all numerical aberrations. Because of the change in the number of the chromosome, this kind of abnormality are happening. Now coming to the types of abnormalities that can occur due to change in the chromosome structure. So one such syndrome is called as the DJOD syndrome. So here it is occurring because of the deletion. Okay, so this part actually it is deleted. Here this part it is actually deleted commonly. So this is the 22q112. That means it is happening in the 22nd, the long arm of the 22nd chromosome in the first region of the first band of the subband 2 is deleted. So because of that, it might have certain genes which are responsible for development of these characters. So if any person is having a deletion in the 22nd chromosome, they will have these kind of abnormal structures, and because of that this will be called a DJOD syndrome. Similarly, I think I have already mentioned regarding the Cry-Ducat syndrome yesterday. It occurs because of a deletion in the short arm or the P arm of the chromosome number 5. Okay. So usually the speech and the language characteristic will change. As I had already mentioned, this region contains genes which are responsible for development of the larynx. Okay, so because of the loss of the larynx, the voice box is the development of the voice will be interpreted, or it will be actually what we can say disturbed. Okay, somehow it will be disturbed, and because of that, the child will have a abnormal sound like the mewing of a cat. Then there is this Williams syndrome that is also occurring because of the deletion. Okay, so before deletion, this is the chromosome, and after the chromosome has been deleted, the size of the chromosome has reduced drastically. So this occurs due to a deletion in the 7th chromosome, the long arm of the seventh chromosome, the first region of the first band of the second 23rd subband. And these are the abnormal conditions that will develop. So like that, in your material you will find a lot of more syndromes. So just remember which kind of syndrome is happening because of deletion or duplication of which chromosome. Like here there is another example, Jacobson's deletion syndrome, that is occurring because of the deletion in the chromosome number 11. Most of the deletions are happening in the long arm, but the Cry-Ducat, it is the short arm or the petite arm. Okay. The 5th chromosome, the short arm. Now coming to something what is unique called as a Robertsonian translocation. So translocation we have already discussed that there is exchange of parts between the non-homologous chromosomes. Now, sometimes what you can observe, two acrocentric chromosomes like this. Okay, these two are acrocentric chromosomes because they have means almost very short arms and quite or relatively much larger long arms. Okay, the chromosomes are here are chromosome number 14 and 21. So two acrocentric chromosomes, sometimes they fuse at the centromere to form a hybrid metacentric chromosome like this. Okay. So this kind of translocation is called as your Robertsonian translocation. And usually the fused short arm of both the chromosomes is lost or degraded due to instability, and this can actually create a problem during the gametogenesis. So we discussed that the Down syndrome, the 21st Down syndrome, or the trisomy 21st Down syndrome is occurring because of non-disjunction during the meiosis. But sometimes the Down syndrome can also develop because of this Robertsonian translocation. I hope the picture is clear to everyone. Okay. So now, if there is a parent who is having a hybrid or Robertsonian translocated 21st chromosome like this. So phenotypically this parent will be normal because it is containing 2 copies of the 14 chromosome and 2 copies of the 21st chromosome. Although one of them is in a hybrid condition, but still every chromosome, that is both 14 and 21st, both are in diploid condition. Now, what are the possible segregations of the chromosome during the meiosis? Now these two normal 14 and 21st chromosome can move to one gamete. Then this hybrid chromosome will move to the other gamete. So this is one possible combination. Or other possible combination is what? This normal 14 and the hybrid might move to one gamete, and this 21 will move to the other gamete. Similarly, this normal 14 chromosome can move to one gamete, and these two, the hybrid and the normal 21, can move to the other gamete. Now, when such type of gametes will fuse with the normal gametes, so what will be the consequences in the zygote? Now, the first combination is absolutely normal, okay, because it will have 2 copies of 14 and 21 chromosome, and when this will fuse with a normal gamete, this will be also phenotypically normal, but again it is a balanced carrier because it still carries this fused, or we can say hybrid chromosome. Now here if you observe, there will be problem, or there will be trisomy 14, because if you observe carefully, there are 2 copies of the one copy of the 14 chromosome is given by the normal gamete, and this gamete is having already 2 copies of the 14 chromosome. So this will lead to trisomy 14. But this may not be that much harmful, and somehow the gamete might survive. And here, whatever gamete will fuse with this one, there will be absolutely a loss of 14 chromosome. So this is monosomy 14. This monosomy 14 chromosome, this will be also means it will also disintegrate or will die. Now, if you consider this condition particularly, okay. So in this condition, already there are 2 copies of the 21st chromosome, and a third copy will be provided by the normal gamete. So this fetus or zygote will develop the trisomy Down syndrome. Okay. So some Down syndrome can also occur because of this Robertsonian translocation between the chromosome number 14 and 21. Okay. So most of the trisomy will occur due to non-disjunction in either of the parent, but sometimes it can also occur because of this Robertsonian translocation condition. And this monosomy 21 will also be disintegrated. Then coming to another concept called as the sex reversal syndrome or the Swyer syndrome. So here, what can happen? Sometimes we have discussed that this SRY is essential for the maleness in case of human beings or in case of any mammal actually. So here, sometimes this Y region can be deleted and it can be translocated to a homologous region on the X chromosome. So now, although this is having 2 X chromosomes, but because of the presence of SRY gene, it will also develop as a male. So this will be a XX male. So although the XX should be regarded as a female, because of the translocation of the SRY region to the X chromosome, it has reversed the sex. That means it has become a male, excess male. Similarly, if in the male, this SRY region will be deleted. Okay. Then what will happen? The XY, in spite of having the Y chromosome, due to absence of the SRY region, it will develop as a female. Okay. So this is what is called as the sex reversal. Being a XY, it will become a female due to absence of the SRY region, and being XX, due to presence of this Y region because of the translocation, it will develop as a male. Then coming to some other abnormalities due to the chromosomal aberration. So here is one example of the retinoblastoma, that is actually development of cancer in the eye. Okay. So here, this RB gene is actually a tumor suppressor gene. So normally it allows for normal functioning of the cell, or it controls the cell cycle in a regulated manner. So because of mutation, if one of the retinoblastoma gene is lost, then also it will develop into a normal individual, because one copy is there to compensate for the function. But usually, what happens if one of the RB genes is lost, there is a tendency that the second RB gene will also be lost. And in this case, being a recessive trait or being a tumor suppressor gene, loss of both copies of the RB will lead to the tumor formation in the eye, or what we call as the retinoblastoma. Then certain other problems will arise due to chromosomal aberration. Okay. I'm so sorry. I couldn't see your message. Is it visible now? The screen visible to everyone?
Yes, ma'am.
Uh, Anyone who is having problem with the screen? So I think I have already suggested you, if you are having the problem, just log out and then log in again. Okay, then you might be able to see the screen. Okay, fine. So here. So some other problems, like I discussed yesterday, that how due to certain kind of chromosomal alterations or aberration like your inversion or translocation, it might lead to cancer. So one such example is this acute myeloid leukemia. Okay. So here you can observe, this is the chromosome number 16, and there is the region in the P13 region, that is the short term, in the 13th band of the short term region, and the 22nd band of the long arm region. So somehow this part is actually inverted. Okay. So because of the inversion, what you can observe, a new fusion protein is produced, and this fusion protein is responsible to push the cell towards an uncontrollable cycle. So if in any kind of stem cell, myeloid stem cell, this kind of inversion is happening, that myeloid progenitor cell will have a tendency to divide uncontrollably. Okay, so that will lead to leukemia or white blood cancer, white blood cell, abnormal white blood cell division, or cancer. Okay. Similarly, here also, due to a translocation that is occurring between the chromosome number 9 and chromosome number 22. Okay. So this is also another interesting that you must remember. After this translocation, this 22nd chromosome is called as a Philadelphia chromosome. So you must remember what is a Philadelphia chromosome, that is sometimes or frequently asked in the question. Philadelphia chromosome, simply I guess it was discovered in Philadelphia. That is why the name is given. So, due to a translocation between the chromosome number 9 and 22, as you can see on the chromosome number 9, there is a gene called ABL1. Okay. And on chromosome number 22, there is a gene called BCR. So these are actually this ABL1 gene is actually a tyrosine kinase, which takes part in the cell cycle or in the cell division process actually. So once this ABL1 comes in close contact with the BCR1, it keeps this gene in permanently switched on condition. So that means the cell cycle will or the cell division will be continuously happening. So that is nothing but your tumor growth, or that is leading towards the cancer. Then I think yesterday also I mentioned regarding the Burkitt lymphoma, but I think I mistakenly told you that it is due to translocation between chromosome number 8 and 11, but it is actually between 8 and 14. Okay. So if there is a translocation between the chromosome number 8 and 14, that will lead to Burkitt lymphoma, means unlimited division or uncontrollable division of the lymphocytes. Okay. So as you can observe here, what is happening? This is the gene, that is a c-MYC gene, that is a proto-oncogene. So proto-oncogene means it has the capacity to induce cancer. So when this is actually on the 8th chromosome, it is silenced. But due to the translocation into the 14th chromosome, this proto-oncogene is activated by the immunoglobulin promoter gene that is present here. Okay, so there will be continuous expression of the c-MYC, and that will push the cell towards uncontrollable division, leading towards lymphoma. So these are some of the human diseases that are occurring because of the structural aberrations in the chromosome. That might be deletion, that might be translocation, that could be inversion. So I hope most of the things are clear till now. If any of you have any queries, you can ask. Then we will move into a very different topic, that is quantitative traits or complex traits. So if anyone is having any question, you can ask now, or we will proceed further. Okay, then let's start with a very new topic, that is called as a complex trait, or in other terms, you can also call it as quantitative or polygenic trait. Okay, so just let me give you a brief reminder regarding the Mendelism. So in Mendelism, what we can say that the traits are actually monogenic. Monogenic means they are qualitative in nature. Like for example, if the genotype of an individual is like this or like this. Okay. So let's say this T stands for tallness in the plant, as we all know. Let's say that T actually makes the plant of height, let's say around 2 meters. So whether the plant has a genotype of TT or TT, every time the plant will grow into a height of what? 2 meters. So this is what is actually monogenic trait. Okay, that means the alleles or the genes are not additive in nature. But in case of polygenic trait, let's say if this capital T is giving the plant a height of 2 meter, and the small t is giving the plant a height of 1 meter. So if it is a more Mendelian thing, or if it is a monogenic trait, then this plant will develop into a height of 1 meter. Okay, this is one. But if for a polygenic trait, what will happen? Now the contribution of each allele will be added. That means for a polygenic trait, this plant height will be 4 meter instead of 3 meter, instead of 2 meter, because the effect of each gene or each allele is additive. Okay. Similarly, this plant will grow into a height of 3 meter, and this plant will grow into a height of 2 meter. So I hope now you understood the difference between monogenic and the polygenic trait. So in monogenic trait, the effect of the alleles are not additive, but in quantitative or complex trait, the effect of each allele is actually additive in nature. So there are two main characteristics. So quantitative traits are influenced by both genetic and environmental factors. The genetic component of the quantitative trait involves the contributions of several genes. So let's see an example here. Okay. So if you observe here, I think yesterday or the day before yesterday, I discussed this part briefly. So you can observe when all the dominant alleles are present, the color is very red because every allele is coding for a little color. Okay, and when none of the dominant genes are present, it is absolutely colorless. And when the F1 hybrid is produced, it is having half of the dominant genes and half of the recessive genes. So the color is actually in between both of these. The color has faded a bit. Okay, and as you will have different combination of genotypes, you can have a series of color like this. So maybe somewhere the number of dominant gene will be 3, somewhere it will be 4, somewhere it will be 5. So if the number of dominant gene is 6, it will be darkest. If the number of dominant gene is 5, it will be little bit dark red. And if the number of dominant gene is 4, it will be light red. And if it is 3, like in this case, it will be medium red. And if the number of dominant gene will be only 2, then it will be much more lighter than this one. And if the number of dominant gene is only 1, then it will be very light in color. And when none of the dominant genes are present, then it will be absolutely colorless or it will be white in color. So here, in if it had been a case of Mendelism, the ratio would have been something different. Okay, but in case of due to the polygenic nature or due to the quantitative nature, the F2 generation ratio is your 1 is to 4 is to 6 is to 4 is to 1 for a case of dihybrid cross. Okay, this is not dihybrid actually, this is a trihybrid. But what I am trying to say is, we know in Mendelism or in Mendelian cross, the phenotypic ratio of a dihybrid test is 9 is to 3 is to 3 is to 1. But if this is a case of your polygenic trait or quantitative trait, instead of 9 is to 3 is to 3 is to 1, the phenotypic ratio will be 1 is to 4 is to 6 is to 4 is to 1. Okay, the F2 ratio will be like this. And in the F1 generation, what you can observe, so in case of monogenic trend, like if we think this capital D, small t, in the F1, all the offspring will be same as the dominant parent, right? But if it is a case of polygenic trait, like I discussed before, the parents will be 4 meter, 2 meter, but all the offspring will be average trait between both the parents, that is 3 meter. So this is a quantitative trait. So often the quantitative traits are coupled with your environmental factors. And what is the difference between the monogenic and the polygenic trait, that you can observe here? If it is a case of your monogenic trait, then as I told, the F1 will be same as the dominant parent. But in case of polygenic trait, the F1 is actually the intermediate character between both the parents. Okay, and always in case of monogenic trait, you will have two contrasting characters, like it is either tall or dwarf. So either the plant will have a height of 2 meter or it will have a height of 1 meter. But in case of polygenic trait, there is a continuity in the character. Like there is 4 meter is there, then slightly less 3 meter is there, then 2 meter is there, then like this. Okay, so there is a continuation of the character. So when you draw the graph against the frequency and the trait, you will have a bell-shaped curve like this for a polygenic trait. Okay. So here also you can observe, this is the corolla length of this particular flower, whatever is given. So in the pure line flower, the corolla length is either this or this. Means it is either short or either, short or either long. Now, what will be the corolla length of the F1 plants? Then the F1 plants will have exactly the intermediate length between both of these. Let's say if this is like, say, length is 2 meter, this is 4 meter. So this will be mostly will be around somewhere 3 meter. And in the F2, there will be more gradation will be there. Okay, let's say like some point means here after, let's say from 2.5 to 3.5 meter. Then again in the F3, there will be more around 2 meter, or more will be around 4 meter. So like this, variations you can observe. So why these variations are happening? The variations are happening because of a combination of genetic as well as environmental factor. Okay. So here if we say they are pure line means they are genetically identical. The alleles, like here let's say it is TT and TT. So whatever variation you are observing here, it is only due to environment. Maybe somewhere the sunlight was more or the nutrition was more. That is why they become longer. Wherever the sunlight was less or nutrition was less, they become smaller. And here also, all of them, they are genetically identical, all the plants, because they are having either capital T or small t. Okay. So that is why whatever variations you are observing in the corolla length, it is only due to the environmental factors. But now here, since different combinations of genotype will be there, so whatever variations you are observing, it is both due to genotype as well as due to environment. Like here if you see. So here now in this case the genotype is identical. Okay, all the plants will have the same genotype, so they will have the same phenotype. Here also all the plants will have the same genotype and they will have the same phenotype. If you observe any variation, it is due to environmental effect. Here also all of them, they are having the same genotype because every plant of the F1 hybrid will have the same genotype. So whatever color variation you will observe between them, it is only because of the environmental effect. But in the F2, there will be different combination of genotypes. Okay. So here, whatever color variations you are observing, it is because of combination of both genotype as well as environment. So these are certain basic statistical concepts which are used in analysis of the quantitative traits. I think most of you must be knowing what is mean, what is mode, variance, standard deviation, etc. So we will use these terms in analysis of our this multiple factor hypothesis or multi-factor analysis of the quantitative traits. So before going into the detail of the analysis, you must keep in mind that we are following a hypothesis called as multiple factor hypothesis, which was given by R.A. Fisher, where a particular value of a quantitative trait, that is the result of the combined influence of genetic and environmental factors. The effect of these factors can be represented as deviations from the overall population mean. So the overall variation that you can observe is calculated by T, which is equal to mu plus G plus E. So mu means this is representing the mean of the population or the mean value of the trait in the population. G is representing the genetic factor or the genetic effect, and E is representing the environmental factor. Okay. So this is how you can analyze. So this is the trait. So trait means the character. Okay, whatever character you are considering, let's say the color we considered in that experiment, right? So like from brightest red to the colorlessness or white color. So usually the intermediate individuals or the individuals with the intermediate character will be highest in frequency. Okay. So this is the mean value of the population. Now, how the other members are differentiated from this? That is actually because of the variation in the, as you can see, there is given G1 and E1, G2 and D2 like this. So why this trait is different from this mean value is because of a difference in the genotype and because of the environmental influence on the genotype. Okay. So coming to the how exactly we can measure it. So there is an example based on which we can predict the total variance is actually a summation of genotypic variance and environmental variance. Okay. So this is the population of a particular wheat plant, I guess. Splice into the overall variation. Where is the plant is given? Yes, this is the wheat. Okay, so the wheat maturation time. This is a population of a wheat plant. Wheat plant. So what we can observe, these are the mean maturation time of different inbred line. That means this is the parental line A, parental line B. So the parental line A have a maturation time of around 55 or 56 days. And this inbred line B has a maturation time of around 72 days. Okay. So, as we know that they are hybrid, the F1 will have a maturation time which is exactly somewhere middle between the 72 and 55. So, that is nothing but your 62. Okay. Now, what is the variation time in the F2 generation? So, it is somehow similar to the F1 generation, that is nothing but your 63. So now if we look into the variance, why or how the individual values or the plants with maturation time are differing from the mean maturation time, the summation of that, it is somehow 1.92. That means every plant in the population will have somehow little bit more maturation difference in the maturation time from one member to another member. So why that maturation time is differing? Now because of the environmental effect. So this is nothing but your VE. Why? Because all the members of all the wheat plant that are in the inbred line A, they are genetically identical to each other. So if the maturation time of this plant is, let's say, 56 days, and if this plant is taking 58 days to mature, the difference is only because of the environment. Maybe it has not got proper nutrition or proper water or proper sunlight, something like that. Similarly, the inbred line B also, so all the plants, they are taking around 70 days to mature, but some might mature as early as let's say 68 days, or some may take time, let's say 74 days. So whatever variation in time, maturation time you are observing, it is again only because of the environmental component, because genetically they are identical to each other. Similarly, for the FE also, for the F1 also, it is only because of the environment. Now in the F2, since there will be different combinations of genotype, this is a combination of both environment as well as genotype. Okay. So how they have calculated it, you can visualize here. So the total variance is actually the average variance. If we take the average environmental variance by considering the first three generations, it is somehow 2.28 days. So simply you can add these to 1.92 plus 2.25, sorry, 2.05 plus 2.88. So together it is constituting the environmental variance. Okay. Now, the average environmental variance we can calculate is simply addition of these three divided by 3. So, what is that value coming out to be? That value is coming out to be 2.828 days. Okay. Now, then how we can calculate what is the genetic variance? So the genetic variance is total variance for the F2 generation. If we want to calculate the total variance in the F2 generation, given is 14.26, and what we found from the previous by analyzing the previous three generations, now the environmental effect of the variance is 2.28. So simply you subtract it and 28. Okay. Whatever value we will get, that is actually the genotypic variance. Okay. So the genotypic variance is only seen in case of F2 generation, because in inbred line A and in inbred line B, there is no variation due to genetic factor, because they are genetically identical. Now, why this calculation is important? Now, this is important to find out a thing, means a term called as broad sense heritability. So what is meant by broad sense heritability? Now, often it is informative to calculate the proportion of the total phenotypic variance that is only due to genetic differences among the individuals in a population, and this proportion is called as a broad sense variability. So the broad sense variability is symbolized by the symbol capital H square. And what is the formula to calculate this? Now, the variance due to genotypic component divided by the total phenotypic variance. So, for this particular F2 generation, if we want to calculate what is the broad sense heritability, we got that the variance due to genotypic variance is, or VG component is 11.98. The total phenotypic variance is 14.26. So, what will be the value? Now, the broad sense heritability will be 11.98 divided by 14.26, which is around 80.84, or we can say 84%. That means among the F2 population, whatever variations you are observing, 84% of the variation is due to the difference in the genotype of the individual, and the rest of the 16% is only because of the environmental variance. So this is what is called as a broad sense heritability, which measures whatever variations you are observing in a population, what fraction or what percentage of variation is only contributed by the genetic component. Then comes another term called as the narrow sense heritability. So the narrow sense heritability is actually the ratio of the additive effect of a gene or the phenotypic variance due to additive effect of the gene or alleles divided by the total phenotypic variance. So again, the genotypic variance can be calculated by considering three components, that is the additive effect, the dominance effect, and the epistatic effect of the genotypic component. So what does it mean? Let me explain you. Like for example, I gave you the this thing. Okay, TT. Let's say there are two genes. So let's say there are two genes that is controlling that color of the image or the color of the wheat, whatever it is. So one genotype could be like this. Another genotype could be capital A, small a, capital B, capital B. Another could be capital A, small a, capital B, small b. Another genotype could be small a, capital B, small b. Okay. So you see, every genotype is different from each other, right? And we are considering that in the quantitative trait, each allele has certain contribution to the phenotype. So when the dominant alleles are more, definitely the character will be more. And if the number of dominant allele is less, the character will be less. So you can observe whatever genotypic differences are there between the members, what is actually contributing to the genotypic difference. Now one is the additive effect of the gene. Okay, like if each A is contributing 2 units and if each B is contributing 1 unit, so what will be the character for the first one? If B will also contribute to 2 units. Okay, because every dominant gene will have the same effect, and the small a is contributing 1 unit, the small b is contributing 1 unit. So what will be the character in the first case? 2 plus 2 plus 2 plus 2, that is 8. What will be the character in the second case? 2 plus 1 plus 2 plus 2, that is 7. Then what will be the character here? 2 plus 2, 4 plus 1, 5 plus 1, 6. And here it will be 1 plus 1 plus 1, 3 plus 2, 5. Okay, like this. So you see there is variations. Okay, so why the character is varying? Now because of the differences in the genotype. So what is contributing to the genotypic differences? Now one is the additive effect of the gene. The other one is the dominance effect of the gene. Because of the dominance, here it is more, and because of recessive, here it is less. And another one is epistatic. So usually epistatic can be, we can say, we can ignore it because we cannot actually quantify it. So qualitatively, maybe one gene will be inhibiting the expression of the other. So that will also contribute to the variation in the character. So, among the total phenotypic variants, only what part is contributed by the additive effect of the gene is called as the narrow sense heritability. Okay. So, of these four variance components, only the additive genetic variance, that is the VA, is useful in predicting the phenotypes of the offspring from the phenotypes of their parent. So, this variance is a fraction of the total phenotypic variant, which is called as narrow sense heritability. So, whereas the broad sense heritability considers the overall genotypic contribution towards variance, the narrow sense heritability is only considering the additive component of the total phenotypic variance. So, it is represented by the letter small h square. So, like the broad sense heritability, the value of h square might lie between 0 to 1. The closer it is to 1, the greater is the portion of the total phenotypic variance that is additive to genetic variance. And the greater is our ability to predict the phenotype of an offspring. So, okay. So, here is one calculation that we will go through. Okay, we can go through this. We can look at this graph and then we can discuss. You can read the text later. Okay, so narrow sense heritability, what we will see here, this is actually a trait, or we can say this is actually a graph representing the IQ. Okay, IQ, intelligent quotient of a parent and the offspring. Okay, so here the mother is having a high IQ of around 110. The father is having a IQ of around 120. So, what should be the IQ of the offspring? Now, it should be somewhere near 115, because we are predicting that in the for a quantitative trait. So, IQ or intelligent quotient, it is actually a polygenic or a quantitative trait. So, here the offspring should have a IQ of around 115. Okay. But what is observed is the actual IQ of the individual or the offspring is 106. Okay. So, which is more than the mean population IQ, but that is less than the IQ of the both of the parents. But if you observe, this IQ is somehow close to the parental IQs. Okay, that means it is not very far from the parental IQ. So we can conclude that the narrow sense heritability in this case is actually will be around 40%. Okay, the 40% it is predicted that the 40% of the character or the intelligence that is seen in case of the offspring, it is contributed by the parent. Okay, and the rest of the variations, that whatever you are observing, it could be due to the environmental effect. So why? Because after you, when you will go through that example, you will get to know that after the birth of the child, they had given the child to adoption. So that means the child is growing in a different environment from that of the parents. So definitely there is some kind of environmental influence is there. But in spite of having the environmental influence, the child or the offspring is having somehow close resemblance or close IQ value towards the parental IQs. So that means some portions of the IQ is definitely contributed by the additive effect of the gene or the additive effect of the genotype that is coming from both the parents. So that is how the narrow sense heritability is used. And what is the use, you might think, why the narrow sense heritability needs to be calculated and known? Actually, it can help in the artificial selection. Okay, so artificial selection means when you are in a from a population, you are choosing some some individual to propagate. For example, mostly in case of plant breeding, we can say. Okay, so in plant breeding, always we want to choose the plants with better qualities or the individual that have good phenotype. Okay, so in that case, this narrow sense heritability is used for selection. So which kind of offsprings you should be selecting, you can predict it on the basis of this narrow sense heritability. Okay, so there is a formula that you can see: R is equal to H square into S. So this is we can put this even in the simple terms if we denote the response to selection by R and the selection differential by S. So how you will be means putting the selection reference now by multiplying the narrow sense heritability with the selection differential. So this multiplication will give you an idea which offsprings you should be selecting to propagate further. So that is actually the utility of narrow sense heritability. Okay, I request all of you to go through the text and put me some question if you are having any confusion or doubt, because we are actually running out of time, so it's difficult to explain this in more detail. So I request all of you, please go through this particular part, and if you get any queries or questions, we can discuss tomorrow. Okay, you can ask me. But the overall what I want to say is, or what I want to convey is, the narrow sense heritability is actually a measure to predict or to select the offspring that you need to propagate for further, because they will be carrying the good phenotype or they will be carrying the important genetic contributions from both of the parents. Now there is the identification of the QTL or the quantitative trait loci. So there are many techniques that are used to identify the location of the genes which are contributing to particular trait within the within the population. Like for example, so what is the uniqueness regarding this quantitative trait is, for example, for a particular phenotype, the genes that are controlling the phenotype might be present on multiple chromosomes at multiple locations. Okay, like this is chromosome number 1, this is chromosome number 2, this is chromosome number 3. So the chromosome 1 is having, let's say, 2 genes, the chromosome 2 is having 1 gene, the chromosome 3 is having 2 genes. So all of them together will be controlling one phenotype. So how you will be locating or how you will be identifying where exactly the genes that are controlling your phenotype are located is called as the quantitative trait loci. Okay. So there are modern molecular techniques that have made it possible to search the genomes for the QTL. So these loci have been identified and mapped on specific chromosome in the model laboratory organisms such as fruit fly and the mouse. And in agriculture, culturally significant plants such as corn and rice. In livestock like pigs, cows, and humans also, it has been identified. The traits have been studied include bristle number in the fruit fly, the obesity in the mouse, the crop yield in the rice and corn, the milk production in the dairy cattle, etc. And in case of humans, such as diabetes, cancer, cardiovascular diseases, and schizophrenia, etc., they can also be identified, the genes that are responsible for them. Then coming to a certain heritability of means certain qualitative traits like the intelligence and personality in humans, how exactly they are herited. So we are saying that the intelligence and the personality, they are also polygenic traits. That means the development of the intelligent quotient or IQ and the development of the personality of an individual could be a combination of both, could be a combination of genetic as well as a combination of the environment. Okay, so it's like if the parents are clever, definitely some parts of the cleverness will be inherited from the parents to the offspring. Though the environment will play an important role in modifying or developing the trait, but still part of the trait will be inherited through genetic component. Like for example, if a child is there and he is getting pretty good education, or he's grown in an environment where having means good exposure and good facilities, then definitely his or her intelligence will be higher, in contrasting to to a child who is growing in a poor environment. But it is not only the environment which will be shaping the intelligence or the personality of the individual, rather it is actually a combination of both. So these things have been established by the twin studies, okay, where the monozygotic and the dizygotic twins have been studied, and based on the study reports, it can be predicted or it can be concluded that the human traits like the personality or the intelligence, etc., they are actually multi-factor traits or complex traits that are a combination of both genetic and environmental factors. Okay. So this is all I have from my side. And regarding um, okay, this thing I have already. Okay, I request all of you to discuss this thing with your regional center or whatever official communication you are having, or mail or anything. We are not allowed to means do anything in this matter, like how to communicate and all. We are just assigned a class. The timing has been assigned to us. We are supposed to come in that time and take the class. And how you will be communicating and regarding your doubt clearing and whatever problems you are having, kindly communicate it with the official team. Okay, I don't have any idea regarding that. I think repeatedly I have told you people, so just request them for creating a common platform where you want to further discuss something or you want to ask your questions or anything. We don't have any idea regarding that. And regarding your query, maybe Priyanka or something, are you still here? Priyanka Das. Okay, I think I have already told you yesterday that the regulation part, the regulation in eukaryotes and prokaryotes, that unit 8, whatever you are talking about. See, I have briefly included that part because there is nothing much to elaborate. You can, if you go through the material, you will get to know. If you come across any doubt, you can ask me. Whatever portions were needed to be discussed, I already included them. I think I told you yesterday also. The part of the eukaryotic regulation is actually the two epigenetic regulators are there. One is the histone acetylation and DNA methylation. So that is how, and the other transcription factors are also there. I told you yesterday. So that means I think I told you yesterday also that these are the ways by which the eukaryotic gene expressions are means broadly regulated, the epigenetic modifications, and in addition to that, the transcription factors. I think we discussed that yesterday also. Means there are variation transcription factors which will also regulate the expression of the gene profile. Okay, so both in eukaryote and prokaryote. So you go through the material. If you come across anything in particular, please ask me. So is there any other question from anyone else, or we can wrap it up? And regarding the assignment, I haven't seen the assignment. The assignment, see, I think I already discussed this also. Always try to means look for the content in your answer. Whatever question is being asked, try to keep that answer within your writing. Okay, so that is what they will be expecting. If the question is for a long format like 10 mark or 20 mark something, make it a little bit elaborate, put the figures and make fragments within the question, like if some mechanism is being asked. So for that you have to first write what are the components of the reaction, then how exactly the reaction is taking place, and what are the conclusions. So in this way you prepare sub fragments of the question, and then you summarize it or you just combine it to represent your answer. And for short questions like 2 marks, 3 marks, be very brief and just put down or select that text which is exactly having the answer to that particular question. Okay, like for example, if I'm asking you your name, so I must look for your name within your answer. You need not include your father's name or a parent's name or the name of your children. So that is not needed. Okay, so be very brief, but always try to put whatever content should be there in the answer. Whatever question is being asked, that answer must be present in your writing. Any other query or any other question, or we can wrap it for today. And for tomorrow, since tomorrow will be my last session, so I request all of you to go through all the 3 blocks. And if you have any question, you can ask me. I might take 10 to 15 minutes more for you to solve your queries. You can go through the material and you can ask me. Or I have already shared the PPT with the headquarter group, so you just request them to share it with you. I don't know if we are allowed to do so or not, so that is why I'm not sharing it directly with you. Okay. Okay, fine. So thank you so much for your patience. And if you have any, okay, see, regarding the assignment, I don't have any idea. Okay, you just have to inquire with the official people. They can guide you. I don't have. They might have some instructions. Go through that instructions before writing the assignment, and if no instruction is there, you can request them to give you a guideline for writing the answer. Okay, so that will be better. You just request the headquarter team that please provide us with a guideline how to represent our assignment. It will be easier for us. Okay, then. Thank you so much.